All right, so this chapter is going to be all about fungi. Um, Mushrooms are your classic example of fungi. They are very diverse, they're widespread, and they are actually very, very important in our ecosystems because they help to break things down and recycle the nutrients that other living things need to be able to grow. There's about 100,000 species of fungi that are around or that have been described, but there's actually about 1.5 million that are estimated to live. Pretty insane. Okay. They do share some key traits in that they are heterotrophs. They get nutrients outside of their body and they use enzymes to help to break those down into the compounds that they need. They act as decomposers, they can act as parasites, and they can act as mutualists. Body structures that we typically find for fungi are as either single cells or multicellular filaments. The single cell ones are going to be your yeast, and some can grow in both forms. Um, because of their structure, they are able to absorb nutrients very effectively. They consist of the mycelia, the hyphae that are able to absorb materials pretty well. The mycelium structure is able to maximize that surface area to volume. We talked about that a little bit with cells so that it can take in what it needs as effectively as possible as opposed to plant cell walls, which are made of cellulose. We talked about bacterial cell walls with the peptidoglycan. The fungal cell walls are made of chitin. Um, the hyphae are divided in cells with septa that allow organelles to be able to allow cell-to-cell movement of organelles so that the organelles can move throughout the cells. The coenocytic fungi don't have septa, and they just have a cytoplasmic mass, which has lots and lots of nuclei present. Pretty crazy. Okay, so there's your hyphae. Your mycelium's making as all the hyphae together, and then you've got your reproductive structure on top, which allows those fungi to reproduce themselves. So there's the septate hyphae, where again you've got the septums divvying up the nuclei, and then the coenocytic hyphae. Sorry if I'm not saying that right. They're all just in one happy clump together. So there are fungi that have specialized hyphae called haustoria that allow them to enter the tissues of their host. These typically will result in mutualistic relationships. We see these with plant roots and fungi. Um, you can have ectomycorrhizal mycorrhizal fungi that will form hyphae over roots and grow into the root cortex. And then you can have arbuscular mycorrhizal fungi that will extend them into the cell walls of the root cells and will be able to go into the cell membranes. Why this would be helpful? The fungi can help to give phosphate ions and minerals, and but that they've absorbed or that are byproducts of materials that they've absorbed to be able to grow. And a lot of vascular plants have these. So, how fungi are able to reproduce? They can reproduce sexually or asexually by producing lots and lots of spores. And there are many, many different life cycles that we can run into with this. We'll talk briefly about these, but again, I don't want you to be focused so much on them. You see plasmogamy and karyogamy. I'm going to talk to you about what those words are. We definitely have already talked about meiosis and germination, but we'll definitely touch on those other ones. So the nuclei for fungi are typically haploid. They do form some transient diploids when they're going through sexual reproduction, and that involves the fusion of hyphae from different mating types. And they use pheromones to determine what their mating type is. Plasmogamy is when cytoplasm is joined together from two parent mycelia. Um, when they don't fuse together immediately, but they exist in one place, they coexist in the mycelium, they're called a heterokaryon. And depending on what type of fungi we have, sometimes we pair off two haploid nuclei to a cell, and those are called dikaryotic. Karyogamy is when the nuclear fission takes place. Again, that can happen within hours, days, or even centuries, and that'll produce your diploid cells. Those diploid cells do not last very long. They go through meiosis to make your haploid spores. And these two processes help to increase the genetic variation among fungi. As I said, they can also reproduce asexually. The molds can produce the haploid spores through mitosis and form that visible mycelia. Other things that we can see that go through asexual reproduction as fungi are yeast. They actually bud off as opposed to producing spores. Again, we can have them form in both ways, both yeast and as filamentous mycelia. And there are some we aren't sure of what they go through to reproduce. The ones that we aren't sure of are called deuteromycetes, imperfect fungi. And once they know what their reproductive cycle looks like, they will be reclassified. So there's the reproduction on the left with the orange on going through molding. We can see the budding off of the spores, and we see it on a more microscopic level down there in the bottom right hand corner. So, where did fungi come from? They came from a protist, but they are definitely more alike animals than they are to the plants or other eukaryotic cells. The fungi, the animals, and the relatives that are protist that are closely related to them make up the Opisthokonts clade. They are closely related to unicellular nuclearides, and the animals are most closely related to the unicellular choanoflagellates. So the multicellular features that we see in both animals and fungi came about separately, but the fossils that we know that led to fungi are approximately 460 million years old, the oldest ones that we know about. So, again, the title of this is that the ancestor was an aquatic, single-celled, flagellated protist. All right, so that is where our fungi came from, and way, way, way the heck back, animals and the chytrids and then the nuclearides. Microsporidae are these fungi. They are unicellular parasites. They do have some tiny organelles, but they are not the mitochondria that we're used to seeing. They appear to be at least related to fungi if they aren't themselves fungi. So more work is going to have to be done to determine. It's thought that fungi probably moved to land or colonized land fairly quickly in terms of when the early land plants did, and they formed those mutualistic relationships that we talked about earlier. Again, there are lots of different forms of fungi that are out there. Being able to look at their DNA has helped to clarify what these relationships might be. And again, there's still more research to be done. So those are the four main phyla that make up fungi: the chytrids, the zygomycetes, the glomeromycetes, the ascomycetes, and the basidiomycetes. So the chytrids are found in freshwater and land habitats, and they can have all the roles we talked about previously with fungi. Um, they are thought to have diverged fairly early, and they have flagellated spores called zoospores. And you can see how there's different groups of chytrids coming off of that evolutionary tree. The zygomycetes are pretty diverse as well. These include your molds, your parasites, and some symbionts. Black bread mold is pretty representative of the life cycle that species in this phylum go through. The Hyphae are choanocytic, so remember that's when all the nuclei are kind of just jumbled up together. And the asexual sporangi will help to form those haploid spores. So there's a picture of the zygomycetes life cycle. Sorry, not zygomycetes. Yeah, okay, I'm not going crazy. They're named for their zygosporangia, which are produced sexually. They have karyogamy and the meiosis take place. They are hard to kill off. They are resistant to freezing, they're resistant to drying, they can survive in some pretty wild conditions. Um, The pilibus actually can aim their sporangi towards food sources so that they will be able to continue to survive and thrive. The glomeromycetes initially were thought to be zygomycetes, but their mycorrhizae are a little bit different. They form arbuscular mycorrhizae. Um, the ascomycetes are found in aquatic environments as well as land habitats. They form sexual spores and sac-like asci, which are found in fruiting bodies called ascocarps. They're called sac fungi, and they will vary from simple yeast to more defined cup fungi and morels. They act as decomposers and symbionts. They also can harm plants. They reproduce asexually with the spores that are called conidia. They are actually not found in the sporangi. They are found asexually at the tips of specialized hyphae called conidophores. And another bread mold, Neurospora crassa, is a model organism for this particular phylum. May have heard of truffles before. So Ascomycetes definitely contain some of the fungi that are more edible. Okay, so there's the life cycle of an example of an Ascomycete. And then we have the Basidiomycetes. Um, these would be your mushrooms, your puffballs, your shell fungi, your mycorrhizae, your plant parasites. They have a basidium club-like structure, which is their diploids, kind of represents their diploid stage in the life cycle. They can break down wood. They're also called club fungi. So there's some examples of the Basidiomycetes. They do have a little bit longer dikaryotic mycelium. So when we talked about what they're named for, and they produce lots of fruiting bodies. So these are the mushrooms that you tend to see if you have, like, if you have a mushroom in your yard, you don't just have a mushroom in your yard. They are able to make those fairy rings where they're all kind of coming off of one set of mycelium underground. So again, there's their reproductive cycle. Um, practical roles of fungi. Again, we talked about each of these earlier. We're going to go into them a little bit more. Decomposers, they can break down cellulose and wood, lignin, and recycle those elements. They could be used in bioremediation to help to bring up the amounts of nutrients that are needed in certain projects. They can act as mutualists and provide benefits for the plant ends themselves, as well as algae and cyanobacteria and animals. So we're going to talk about mycorrhizae a little bit more. Um, Plants have these endophytes, which are fungi that can live in their leaves or other plant parts. And these endophytes make toxins that keep herbivores or pathogens from attacking those plants. And the endophytes typically are ascomycetes. Sometimes fungi help out animals by helping them digest things. So cow guts, they eat a lot of plants or other grazing mammals. Fungi help to break things down. And ants can take advantage of fungi by raising them in farms. So lichens actually develop from a symbiotic relationship between a microorganism that's photosynthetic and a fungi. You basically have all these photosynthetic cells that are held together within the fungal hyphae. The photosynthetic is usually from some sort of protist-like structure, green algae or cyanobacteria. The fungal is an ascomycete. And then the symbiosis, how they work together, is so interchangeable that they're actually given specific scientific names. And then the algae or cyanobacteria live underneath the lichen surface. The algae give the lichen the carbon compounds. They also help, the cyanobacteria gives it nitrogen, and then the fungi gives environment for the algae or the cyanobacteria to be able to live. So those are examples of lichens. They can reproduce sexually or asexually. When they reproduce asexually, it's by fragmentation. We're forming ceridia. making small clusters of the hyphae that are embedded within the algae. They are definitely helpful with new rock and soil surfaces, and they may have helped to colonize land um, by plants. And pollution can definitely be a deterrent to them. When they are starting to die off, that's a pretty good sign that there's some issues with air quality. There are many, many fungi that are known as parasites, so you definitely cannot ingest every fungi that you see. Fungi can kill things off, they can kill off plants. Um, and some of the fungi that do kill off food crops are also not are toxic to humans. So definitely need to be careful with what fungi you eat. Um, So this pretty well. Ergots contain lysergic acid, which is the raw material for LSD. Definitely not something you want to take just randomly. Animals definitely are not as susceptible to the parasitic fungi as the plants are. And there's just an example of a fungi that might have led to a decline in a particular animalian species. So those are some examples of fungi that are pathogenic. Or harmful, sorry. And then when you are a pathogen, that's going to be specifically with an animal. If you have a fungal infection, you are typically going through a mycosis. Ringworm, athlete's foot are examples of mycoses. Um, Depending on fungi, because they're so hard to get rid of, when you get a fungal infection, in a human body that can cause lots of issues. Coccidomycosis yields tuberculose-like symptoms, and the fungi can also be a source of yeast infections. So we talked a little bit about why fungi are actually good for us. We eat them. We can also use them to help to make bread and cheeses and alcoholic beverages. They can help make antibiotics, aka penicillin. Um, and they are being used to look at genes that are thought to be involved in various genetic disorders and to grow proteins that will help with insulin. And that is just a summary of the different phyla that make up fungi.